Welcome to the Monday plenary session. Please welcome APS President George Abawi.
Welcome and thank you for joining us at this afternoon plenary session. I would like to extend a special welcome to all those around the globe joining us via live feed, video feed today. The theme of this year's meeting is Plant Health Connections. As I mentioned yesterday during the opening general session, the major goal of the diverse investigation and outreach activity conducted by plant pathologists is to guard the health and productivity of plants. However, guarding the health and productivity of plants require not only understanding the pathogen plant-environment interaction, but also the targeted production system, inputs and practices employed, soil health, and many others. Indeed, all these topics are interconnected and directly or indirectly contribute to the grand challenges of agricultural productivity and sustainability, food safety and security, climate change, environmental quality, and others. In this plenary session, we will focus on the connection, a bit in depth, between soil health, plant health, food security, and human health. Today, I'm truly pleased to introduce three outstanding speakers and leaders in their fields to address these interconnected and impactful topics. Our first speaker is Dr. Harold Van Ness, a professor of soil and water management and former chair of the Department of Crop and Soil Science at Cornell University. Harold will discuss the emerging concept of soil health, assessment protocols, sustainable management practices, and the overall impact on soil functions. Our second speaker is Dr. Jan Leach. She's a University Distinguished Professor at Colorado State University and adjunct scientist at the International Rice Research Institute. Jan will highlight the impact of soil health and management practices on plant health and the role and connection of the phytobiomes to plant health and productivities. Our third speaker is Dr. Jennifer Ann Thomson, a professor of microbiology in the Department of Molecular and Cell Biology at the University of Cape Town, South Africa. Jennifer will be speaking on the connection of healthy plant and food availability on world food security and poverty issues. Her talk is provided through the Glenn Anderson Lectureship on World Food Security Award that is presented at the joint APS-CPS meetings. Complete list of all our speakers can be found in the program book, and I encourage you to look it over because there are a lot more information there. Our speakers will each have 25 to 30 minutes, followed by a facilitated question and answer session. We encourage our global audience to submit questions following the presentation through Twitter. Now, I hope you join me in welcoming our first speaker, Dr. Harold Van Ness, who is a fellow of both the Soil Science Society of America and the American Society of Agronomy. My colleague and collaborator, Dr. Harold Van Ness.
Good afternoon. I hope you're having a great day and enjoying the conference. Thank you, George, for that kind introduction and the invitation. It's truly an honor to be here. I decided to put my assignment that George gave me into a slightly larger context of, you might say, threats and opportunities, climate change, and also the opportunity of data-driven management and communication and information technologies. Also talk a little bit about national initiatives that some of you may be aware of, and perhaps some of you are not, and I think it's important to discuss that. You've all seen all those graphs about climate change, carbon dioxide increases, temperature increases. I thought this one was actually, I hadn't really seen this one until recently, was very interesting, and I think especially in the context of your interest as a discipline. And this focuses on the increases in very heavy precipitation events over the past decades, and you can see that there's a real geographic distribution to it. That the eastern part of the country is receiving more precipitation and more heavy precipitation, so therefore extended periods of soil wetness and higher relative humidity and all those things that are concerned in terms of managing diseases. The Northeast seems to be the worst, and 71% increase in heavy precipitation. That's very significant. I was glad to find these maps, not only because they talk about projected changes, but also because they actually have Canada on it. And I know some of you are from the CPS. I apologize for the previous slide. But this here shows, now these are projections, model projections, of course, but I think that there's an interesting nuance here. So you see that the 4 seasons, and since most crops are grown in the spring and in the summer, let's focus on that. So the southern part of the continent appears to be getting less precipitation as projected, and the northern parts are going to be receiving more precipitation during the spring. And that's not necessarily good, because the parts that are already dealing with more precipitation and excess wetness are going to get more, and the drier regions are going to get less. So that's not good. Now, for the summer, we see that most areas are going to receive, on the average, less precipitation. This is the time of the year, of course, when the crops are fully grown and pumping a lot of water through transpiration through the system, and they're going to be needing that water. So in general, I would say this is not necessarily a good trend. But we're finally getting serious. I think our president is taking on some initiatives. I think because of the gridlock, he's willing to be a little bit more of a leader on this. Several months ago, the Climate Data Initiative was launched that focuses on helping communities, and including agriculture communities, providing with better climate data so that they can do more planning, for example, flooding and all that. And then just 2 weeks ago, the White House announced the Empowering the Agricultural Sector and Strengthening Food Resilience Initiative, which also has a very heavy data component that actually kind of builds on the Climate Data Initiative. Now, for the Food Resilience Initiative, that involves a number of interesting entities. The World Wildlife Fund, Microsoft, PepsiCo, and Monsanto are one of the key players. So this is good news. So with all these climate change challenges, I wanted to talk about some win-win opportunities for both adaptation and mitigation. And there's 3 examples basically from work that's being done at Cornell. I'll talk a little bit about Bill Fry's late blight decision support system, because Bill's team and our team have been working fairly closely together. We've been sharing a programmer, so we've been able to share a lot of ideas back and forth. Then I'll talk about AdaptN, which is a nitrogen management tool for corn, which we developed, and I'll primarily put it in the context of how we use the technology rather than, you know, maybe some of you are not that interested in nitrogen. And then the Cornell Soil Health Management Framework, which is really my initial assignment initially from George. So unless you've been living in a cave for the past few years, you know that cloud computing and new information technologies are changing the way we live and we work. You know, we all have these mobile devices. We have seen a tremendous increase in the use of large data centers, large storage capability, instant communication. And so we have an opportunity to take advantage of that, and I'll show you that through these examples. From a management perspective, we can move from more generalized recommendations to very sort of real-time, adaptive, and site-specific recommendations. So there's tremendous opportunities in that area. And then, of course, you know, we have the communication and convenience functionality that we can build in because of the, you know, these smartphones and tablets and all that have wonderful base software that you can build on. And by the way, a lot of our stakeholders, of course, are highly mobile individuals, farmers and consultants. They're in the field or on the road all the time. So these people actually benefit more from this technology than some of us who are mostly sitting in an office. Both the Late Blight project and the Adapt-N project have benefited a lot from the high-resolution climate data that are now readily available. We're working through the Regional Climate Center consortium, mostly the Northeast Region Climate Center at Cornell. This is some example of high-resolution precipitation data from a day in June in Iowa in 2011. And the main point I want to get across is that, you can see how variable precipitation can be. In one area, they're receiving 6 to 8 inches of rain in this one day, so a lot of rain. But you move maybe 10, 15 miles away from the hotspot, and they maybe only received 1 or 2 inches. So having really good high-resolution climate data is really critical for these decision tools that depend on precipitation information or even solar radiation or temperature information. So I'm going to show a couple of the user interfaces of Bill Fry's Late Blight Decision Support tool. And I believe he told me he gave a workshop a few days ago, so some of you may have seen it. Just want to point out more the functionality rather than the science per se, which is, I'm not an expert in that. But you can, so basically for this tool, you have an interface. It's all located in the cloud, basically, on server space. We both now use commercial data centers rather than the servers at the Climate Center because it's a lot less expensive and a lot more secure. In fact, it's remarkable how inexpensive the commercial data centers are. Amazon is actually the primary purveyor of that. So not only selling stuff to you, but also providing data center services. So for Bill's tool, the primary inputs are, you choose a location, and I didn't show the interface for that. It's basically based on a Google map so that you can access the climate data, and then information on host resistance and fungicide applications. So you select a cultivar and the resistance class, then the pathogen lineage, so that relates to the susceptibility, and then any fungicide applications that have already occurred. And then this tool uses both observed data, precipitation, temperature, and humidity, as well as forecasted data, because it's very important for this particular tool. And on a 2-kilometer grid for the forecast data. And this is then, the simulations run automatically every day, I believe every morning, and then generates an email-based report that you can access then, through any device. The simulations are a function of weather, host resistance, and fungicide. And then you see the report. It actually gives some information that is really quite insightful. So in this case, the simulations were run on May 29th, and so the climate data, the weather data up to May 28th was included, and then you see the several days of forecasted data, both in terms of whether there's going to be rain or whether the relative humidity is going to be of concern. And then indicates the blight units and the fungicide units, and I don't really exactly know what that means, but I think probably most of you do. And then whether there's a threshold exceedance or not, which is indicated in red. And so that should prompt a grower to take action. And so that's the basic idea. So like I said, we've been able to benefit from working together. The Adapt-N tool, however, was licensed and commercialized this past December. So our interface is looking a little different. This is what you get when you bring the pros in. Now, before I proceed, my university requires of me that I show this slide. I have to disclose to you that this technology came out of my program, that it has been licensed and is being commercialized by a startup company named Agronomic Technology Corporation, and that I have a small equity interest in that company, and that company is providing some limited support for research. So, in other words, I'm biased, as if I wasn't biased before, but I have to do this. This is a university requirement. So I'm not going to go into a lot of details about what ADAPT-N does, but it basically uses a little larger set of input data to make a recommendation, and that includes weather, precipitation, temperature, in our case also solar radiation, irrigation amounts and dates, then some soil information, texture, tillage, and fertilizer applications and manure applications, etc. Some basic crop information like cultivar and rotations, etc. And then it has an economic component that includes the relative prices of fertilizer and grain, as well as a profit loss risk factor. And I'm sorry I don't have much time to go into that, because I really want to focus on sort of how we communicate basically with the stakeholders and provide this information. So just a few screenshots. So again, you either identify a field on a map, or you can drop a shapefile if you have a GIS-based system. Then you provide information on the soil type, whether it's drained, artificially drained or not, the organic matter content of the soil, and some basic relevant soil information. Here's one of the screenshots that addresses the crop information. So the maturity class of the corn, the expected yield, the date of planting, the plant population, and then rotational information. Is it corn after corn after soybean, corn after sod, how much legume is in the system, etc. Now you notice on the right side there's some functionality there that makes it easier for the grower. So if you basically use the same varieties and more or less the same planting date and all that, then you just click on each of the fields or zones, and then you apply that to the so it keeps you from having to enter the same data over and over again. There's other input screens as well, and I won't show that. Just want to give you a taste. So this is then the recommendation that comes out of it. So you basically run the tool, run the simulations, and it's all done at this centralized data center, commercial data center. In fact, I don't even know anymore where it is. It used to be at Cornell, but it's somewhere in New Jersey, I believe. So here you see then a typical report. The recommendation would be 140 pounds of nitrogen for this particular field. We give sort of a range to that, some what you might call confidence interval. And then, we provide a lot of additional information. And I think the philosophy is we try to be as transparent as possible. For example, we indicate how much nitrogen is currently in the soil, how much is currently in the crop, how much is going to be needed to complete the crop, in this case 184 pounds. How much precipitation was received between the date of planting and the current date. Things like that sort of provides more insight. The other thing that I want to note is the date selection. So the default is that you run the tool for today's date. So, well, today's date is August 11th, and that's not really a date when you would be applying fertilizer to a corn crop. But if you wanted to go back and say, well, what would the recommendation have been on June 13th or something like that, then you just enter the date and you get your recommendation for then. It's really also very useful for, say, a consultant who wants to sit down with a farmer in January or February and say, let's look at some of the options. What would have happened in the last growing season if you had, say, put less nitrogen on in the early spring and put more on in the late spring. We also give a lot of graphical outputs, and I'll just show you a couple of those. This is cumulative precipitation, leaching, and gaseous N losses. And we feel it's very important to be transparent. Now, of course, you open yourself up to potential criticism because you're really, putting it out there. You're exposing all the details of your tool. Here's another one, mineralized nitrogen, so how much release of nitrogen occurs, and temperature, of course, they are related to each other. So, and we have a number of other graphs that we put out there. And, most of the sort of expert users really appreciate that. And the other thing is a lot of the folks who teach courses on fertility management use this tool, and they find that very useful. Similar to Bill Fry's late blight tool, we have a daily alert system. In fact, I believe we actually got that idea from Bill because it really was very useful for the late blight prediction, but we found that it actually was a feature that many of the users of the tool appreciated very much. So you get an email and within that email you can directly click on particular fields and get the reports and all of those kind of things. And that kind of functionality is really possible now. So every day you would get basically an update on the status of your field, so you can monitor that field on a daily basis. We also, with the commercial version, we brought in basically some functionality that would make it a lot more attractive to a user. So we have a farmer dashboard and we have a consultant dashboard. So for the farmer dashboard, you have some information that sort of gives you a quick overview. So for example, it gives you the maximum and minimum recommendation for any of the fields that are in the system and an average. It also gives you the total amount of nitrogen that's going to be needed to fertilize all of your fields. So the farmer would basically, in this case, have to order 7,120 pounds of nitrogen. So they don't have to multiply the recommendation times the number of acres and all that. It's provided to them. And on the left, you see these bar graphs indicate what the stage of development for all of these fields is. So is the corn at the V6 stage or the V8 or the V10? So it allows a farmer to track that. So these are all basically features of the underlying models, the soil and the crop model, but we're kind of putting it in a format that makes it more attractive to a farmer. And on the bottom part of the screen, you have all those fields listed. You only see 2 here, but you know, a typical farmer might have 20 or something like that. We also have a reporting feature, so it generates a PDF report, very important for both record keeping, but also for the increasing regulatory pressure. So you can really have a record, date and time stamped of what the recommendation was. And so it gives you the recommendation as well as the inputted information. So it's also a very popular feature. So what's the bottom line for these 2 decision support tools, so the late blight tool and the adapt-end tool? Well, obviously, ease of use, transparency, and insights, very important. That also offers incentives for better management. We've seen that by providing the information to a farmer or consultant, they start changing their management practices towards more sustainability. But overall, the bottom line is profit increases and less chemical use, so environmental benefits. So that's really the win-win scenario. And so it's the technology that allows us to manage the disease or nitrogen much more efficiently with really relatively little input. So this is kind of interesting. We commercialized the tool, we licensed it and commercialized it to a startup company. But just in the past year, both Monsanto purchased Climate Corporation, and also DuPont Pioneer came out with somewhat similar versions of the tool. Imitation is the highest form of flattery. And they did have the benefit of looking at our tool and evaluating that and develop their own tool. We're happy to see that the technology is spreading and that there's basically large corporate buy-in. The other thing that's been very interesting, to us at least, is that entities have expressed an interest in this technology that we had never expected. We're working with Environmental Defense Fund and the Nature Conservancy and some other environmental groups, in part also working with Walmart. And this is very interesting. Walmart has a goal to reduce its carbon footprint. And when it did a total life cycle analysis, it found that, and it looked at the entire supply chain, it found that almost half of its carbon footprint was associated with nitrogen fertilizer. So it's not really the carbon, it's the nitrous oxide that is the concern. And so since Walmart has this reduction goal, it is now starting to work on its supply chain to reduce the nitrogen inputs and have more efficient nitrogen use. And Adapt-N is listed as one of their high-level tools that they are promoting. So Walmart is now pushing this up the supply chain. So actually General Mills, which is from Minneapolis, is now initiating in turn a similar initiative. And Walmart is putting pressure on Kellogg, on Cargill, and all those other companies that basically are involved in crop production, and therefore their suppliers, the farmers, are using nitrogen fertilizer. To me, this is just absolutely fascinating, and it's still ongoing. Okay, so that's the decision support tools and how we're using communication and information technologies. Now I'm going to talk about soil health, George. There's two major initiatives just in the past year on soil health. One is the Soil Renaissance movement, which is supported by the Noble Foundation in Oklahoma and the Farm Foundation, and emphasis on measurements, economics, education, and research. And then the second one is the Soil Health Partnership. And this involves the Walton Family Foundation, again, the Walmart connection. Monsanto is supporting this, and it's the National Corn Growers Association that's taken the lead. Howard Buffett also has been providing some support through his foundation. Another one is the USDA strategic plan. Currently actually has soil health listed quite prominently as one of their goals to improve soil health. And the Natural Resources Conservation Service has a major initiative on soil health management. And it's interesting, we've been working in this area, George, for 10, 15 years probably almost. But once the federal government decides that they're serious about something, boy, things happen. Money flows and staff is reassigned, and it's really wonderful to see this happen. Now here's my comment. I don't believe that this community, the phytopathology community, is very involved in these initiatives. And I may be wrong about that, but I'm just saying. Okay, I'd be happy to work with you in terms of, because you are a very important player in this, and I think you should probably have a higher level of involvement. Okay, so let's talk a little bit about soil health in general. So here you can see two soils. It's basically the same soil type. It's a Buxton silt loam in the northeast. And you can look at the one on the right and one on the left, and I think most of you have seen soil like that, and you can make some interpretation. Now, if you use a standard soil test, which is really a chemical soil test, pH and nutrients, according to that test, the one on the right is a better soil than the one on the left. Okay, it has better chemical features. But of course, what's missing is that we're not really measuring anything related to the physical or the biological aspects of the soil. And so when we started our soil health program, and George was really leading that, we decided that we wanted to expand the traditional soil test, which is really highly successful. When you think about it, a typical farmer knows how to use a soil test. They submit samples annually or biannually maybe, and they get the results. They are familiar with the numbers. They know how to interpret them, and they know how to act on them. Okay? So we said, well, but they don't know anything about physical or biological functions in soils, so let's develop a test that addresses that. All right, so if we think about the three realms of soil health, the physics, the chemistry, and the biology, and these are some of the processes that are critical. For example, aeration is a very important process related to some of the things that you are interested in, water storage and movement, etc. So we're not really offering any testing services to address the physics and the biology of the soil, but we have, of course, for the chemistry. So, and we felt it was important because if you don't know if you don't measure, okay? Or what gets measured gets done. These are sayings that people use. And it's true. If you never measure your blood pressure, right, you don't really know whether you have a problem or not, okay? So if you never measure your temperature or any of those things, so it's really important to become a little bit more quantitative, and because that allows you to manage it better. So one of the challenges that we had was, well, how do you select indicators? We all have books on our shelves that are really fat and that have all kinds of methodologies to measure all kinds of soil indicators. But we needed to set some criteria, okay, for these new physical and biological indicators. It had to be standardized, it had to be consistent and reproducible, it had to be relevant from a scientific and agronomic perspective. It had to represent all the diverse processes in the soil, the previous slide. You had to have something for aeration, something for nitrogen mineralization, etc. And it had to be relevant to agricultural management, not just so that a grower could actually act on that. The other thing that was critical in terms of adoption was that it had to be relatively easy to do and inexpensive to measure. Interpretations needed to be accessible to many users, and somewhat minimal infrastructure and investment. There wasn't necessarily a whole lot of money available to do, to set up labs and all of that. Now, keep in mind that we did this during the genomic revolution, and that's where all the funding went at that time. But as an example, in terms of being easy to measure, we initially had included bulk density as a variable, but it required sending out rings and all that. And we found that most people can't really take a good sample for bulk density. And so we had to nix that and say, okay, we're just going to take disturbed samples and that will be fine. This is basically, these are the indicators that we selected based on those criteria. So there are physical, biological, and chemical indicators. So the physical ones are available water capacity, surface and subsurface hardness, aggregate stability, very good indicator, by the way, organic matter, soil protein, root pathogen pressure, respiration, and active carbon. And then the chemical indicators are basically our standard soil test. So we didn't really mess with that. Now, one of the things that you might notice here, and this is sort of a typical soil test result, is that the chemical indicators were all green and they all scored 100. Okay, I'll talk about how we come up with these ratings. And that's because, farmers know how to test their soils for pH and nutrients. They never tested their soils for the biology and the physics, and so you see that there were some real problems with that. You see a lot of red and some yellow. Okay, so that's, and I'll come back to that, but if you don't measure it, you can't manage it. So again, one of the key objectives was that for all the important processes and functions of the soil, we wanted to have at least one indicator that could represent that. So you had something for aeration, something for infiltration, something for root penetration capacity, something related to root disease pressure, which was really George's main contribution. Okay, so it's important if you want to have a comprehensive test that you have good indicators. Just going to show you a couple of slides of, and the details don't matter as much. We measure aggregate stability using basically a rainfall simulator. We see how well the soil holds up against rainfall energy in sieves. Active carbon is a very simple test. Active carbon is basically that part of organic matter that's biologically active, so it's being turned over. And we just use a simple permanganate oxidation colorimetric test. I think it's certainly less than $1,000 of investment. We rolled out a protein test just this past year. We felt that was actually a very important part of the soil organic matter. We also measure respiration, basically an indicator of the abundance and metabolic activity of the soil. And I put the picture of the Earth from space at night, and you can kind of see where the abundance and the metabolic activity is. So it's kind of the same idea, okay, just to kind of put that in your mind. Okay, well, this is a slide from George, and I think it's a familiar concept, in plant disease management. So, we're dealing, of course, with concerns about pathogens, and so in our test, we're not being specific to any particular agent, but we are looking at sort of an integrated pathogen pressure assessment, and of course, the environment is important as well. That kind of gets us back to the climate change and more precipitation, more extended periods of high humidity, etc. And then a susceptible host. Now, I think when we think about the interactions with soil health, then let me actually go back real quick because I just showed you this on the environment. I'm showing this very compacted soil. So a lot of these physical indicators, as well as chemical indicators, really affect the potential for expression. So if you have a very compacted soil, remains wet for extended period of time, very low aggregation, very low aeration, you tend to have more concerns with certain pathogens like Phytophthora. Also, pH, of course, is important with certain pathogens like scab. And so that's the physical and the chemical is kind of maybe a little bit more apparent. The biological, of course, there's some very interesting. It's a jungle down there, and if you can create an environment where you have more suppressive capability, where you have competing organisms competing for space or food, or they all eat each other and get eaten and all that. But having a more diverse community is attractive, and I think you are probably more familiar with that than I am. So supporting that healthy biological systems is important. This is the bioassay that really came out of George Abowie's lab, and this is his slide. So basically, what we do when the samples arrive and we do all these analysis, part of the sample is being used to put in these growth tubes. They go into a greenhouse. We seed a very susceptible dry snap bean, and then we grow it for about 4 weeks. We wash the roots, and then we do an assessment of those roots, which George gave us all kinds of guidance on. And so this is now, since 2006, actually, this is a practice. We receive samples and we do these analyses. So this is also, we basically then, that ends up being going into a rating. So rating of 1 or 2 is very good and gets the green, and then a high rating of, say, 8 means that it's poor. And George has specific things that we look at, lesions and coloring and all those kind of things. So let's go back to the test report and talk a little bit more about how we sort of develop this framework. So we have all these indicators like available water capacity and all that. Thank you. So we have a measured value that comes with that, and then we have this rating, and we then give it the familiar color scheme of red, yellow, and green. We also then list what the specific constraints are associated with that. So we do that through so-called scoring functions. So a value of 210 for active carbon, is it high, is it low? We didn't know, really. We knew pH, we knew maybe phosphorus, but some of these indicators, we really had no idea what was a good value or a bad value. So we have 3 types of scoring functions. More is better, like, say, organic matter or aggregate stability. Less is better, like penetration resistance, hardness of the soil, or actually George's root ratings, or an optimum, pH being the obvious example, also phosphorus. You can have too little or you can have too much. So how do we then develop these scoring functions? Well, for the cases of the indicators where we didn't really have a good sense of whether it was a good or a bad value, we just sampled a lot of fields, mostly vegetable, but also field crop fields. And so some of we knew were really bad soils. They'd been poorly treated and intensive tillage and no good rotations and blah, blah, blah. And then some were very good fields, that had been really built up. And, I've been part of organic production systems, lots of inputs and all that. And then basically, then we had a distribution. And in almost all cases, that was basically a Gaussian distribution. And so very simple to fit. You have 2 parameters, a mean and a standard deviation. And then you take that Gaussian, that bell-shaped curve, and then you make it into a cumulative curve, which is the cumulative normal distribution function. And that looks like this, and it looks like a nice scoring function. Okay, so on the left you have the fields with poor health, and on the right the ones with very good health. And so this cumulative normal distribution function, which is listed here, still has the same 2 parameters, the mean and the standard deviation. So once you define the mean and the standard deviation of your distribution, then you have a scoring curve. We separated it out by textural class because that does matter in most cases. And then you see also where the interpretations are of what's red, what's green, and what's yellow. Okay, so it's a relatively simple system to implement. So we did this for the Northeast. If you want to apply this to South Africa or China, then you can develop your own scoring functions relatively easily. Okay, so that's how we came up then with these ratings. So it has the color, and it also has basically a score between 0 and 100. So then the next step was, okay, that's nice that you have a report. So what do? What are the actions that a grower could take? So we developed a number of management recommendations for each constraint. Okay, so for example, if aggregate stability is low, then some of the recommendations are incorporate fresh organic material, use shallow-rooted cover and rotation crops like, say, a ryegrass or something like that, and maybe add manure, green manure, or mulch. And then we also had some long-term recommendations, like, for example, reduced tillage, which really takes a little bit longer to implement. So for each potential constraint, we have a set of management recommendations. Okay, so, and then, all right, then you have some recommendations, and so how do you then get to the next level? So let me step back a little bit, because, you know, we really learned that you need to sort of indicate to the user what this report is for. First, it's a management guide. It's not a prescription. People are used to soil testing being a simple prescription. Okay, if your pH is 5.6 and it needs to go up, you apply this many tons of lime, or you apply this many pounds per acre of phosphorus fertilizer. When you look at the more holistic management, it's not that simple. Okay, and as an analogy, let's say for your human health, you've measured your blood pressure, your heart rate, and you had a blood test done, and you have a cholesterol scan, and you have some problems. You have multiple options. You know, maybe you want to just simply take a pill. Maybe you don't want to take Lipitor or something like that. But maybe you need to lose weight. Maybe you need to exercise more. Maybe you need to reduce stress in your life. And so what works for any particular individual may vary, okay? So also, you know, you have these different management options. They may address the same problem, or vice versa. You can have one management practice affect multiple constraints, and I'll show you an example of that. And keep in mind that soil health changes slowly over time. It's not like, you know, you're low on phosphorus, you apply 50 pounds, and your problem is solved. Oops. So here's just an example of that. One practice can address multiple constraints. Depending on what cover crop you select, and it's a legume or a grass or whatever, there's a whole bunch of potential benefits that you may have from that single cover crop that may address one, two, or maybe more constraints that have been identified by the soil test report. But you could also have one particular problem being addressed by multiple practices. Okay, so how do you sort this all out? So we actually, what we found, and George probably remembers this, that initially when we rolled out the test, that our the extension field staff and consultants really had problems, not with interpreting the report, but getting from that interpretation to actually suggesting management practices. So what we decided was to actually just develop a 4-step, which is now 6-step approach, that just kind of parsed that out a little bit and made it easier for people to make those changes. So and they're listed here. It's basically you take information from the report, you identify the management options, you bring in kind of local issues such as, you know, what's the financial status of the farm, what kind of equipment is currently available, what's the attitude towards change of the farmer, and some of them may vary a lot. Some want to be on the bleeding edge, some of them are very conservative. So these were all, and I won't go into details, but you know, we realized there was really a missing component because we were in our own scientific world. We thought we had done great, but we found that people had problems going from the report to management practices. Okay, and I wanted to just, this is my second to last slide because I wanted to mention this to this audience. So George's bioassay is really great. It's a very good integrative assessment. But it's also somewhat generic, and it's relatively time-consuming and relatively costly. So it's a very important part of our test. But we're challenged by it, in part also because of the time requirement. You know, it really delays our ability to report because we have to grow the beans. So it would be desirable to have the tests that are more rapid, more agent-specific, and less costly. Okay, so again, oh, there he is again. Just saying, I mean, I'd love to work with people to improve that aspect of the test. So here's our website. If you're interested, if you just Google Cornell Soil Health, you'll get that. We have a wonderful training manual that actually Beth Gugino, who's now at Penn State, took the lead on. We're now working on version 3. Actually, starting tomorrow, we have a large international workshop at Cornell, and I need to leave for that right away. And so there's a lot of interest, and I wanted to acknowledge these people here and these funders. Thank you very much.
Thank you very much, Harold. Well, we better move on, and I hope you please join me to welcome our second speaker, Jan Leach, who doesn't need introduction to this audience, but Jan she's a fellow of both APS and AAAS. She was a past president of APS, and she's the current director of our Public Policy Board.
Well, good afternoon. It's my pleasure to be here to talk to you today about a new initiative that the Public Policy Board is promoting for our society, the American Society for Plant Pathologists. So today I'd like to start with an acknowledgement that this initiative bubbled up from a thought process that was pulled together by Mike Boehm when he was president of APS, and it has emerged into this topic that we're calling phytobiomes. So it's a process that came through the membership and now is being honed into a process of policy advocacy. So what is the phytobiome? Well, if my SWAT teams from the Public Policy Board have been doing their job, you all know what the phytobiome is. The phytobiome is the entire microbial community in, on, and around the plants. So tops of plants, bottoms of plants, roots. And this community is not only inside the plants, the endophytic compartments, but also outside the plants, the exterior of the plants, the rhizosphere. It is the community that influences the plants or that is influenced by the plants. So it can lead to better nutritional qualities of the plants, but also healthy plants or more food, feed, and fiber. It includes bacteria, archaea, fungi, oomycetes, nematodes, and other living organisms that surround the roots and influence the plants. So you can see that now we're looking at a more holistic, system-wide scheme. The initiative, the Phytobiomes Initiative, is meant to target an understanding of how that associated community influences the plant or is influenced by the plant, and how we ultimately can use that information to improve crop productivity, quality, and safety. So the goal of this initiative is by 2025 to have a comprehensive understanding of phytobiomes so that we can accelerate crop production and improvement. So where are we starting? And I like to think about this as the old story of the blind man and the elephant, where historically we've looked at individual compartments or individual, like the rhizosphere, or we've looked at the leaves of the plants, and we've interpreted the plant and the process, or the phytobiome as we now are calling it, as either what organisms are associated with the surface of the leaf or those that cause disease. But really, it's not just the individual organisms. They act as communities and they act as groups, and they influence the plant in that way. So historically, while we've only been able to look at one or a few organisms at a time, now we need to start thinking this as a system. What about the communities of the organisms, and how do they influence not only the plant, but other microorganisms in that community? This, of course, includes cultured and non-cultured microbes, because we've not been able to look at those uncultured microorganisms before, and yet we knew and we know now that they do influence the health of the plant, the health of the soil, and the health of the environment. We're also interested in how the whole system works. Before we've looked at how a microbe causes a disease, or a microbe benefits a plant by acting as a biocontrol agent. But those organisms function together, the plant and the microbes, differently under different environmental stresses. So what happens? How does drought factor into this? How do high temperatures factor into this story? And these are questions that historically we've not been able to address holistically because we haven't had the tools to do so. So can we integrate this whole system to understand it? Well, our question of why are we looking at the phytobiomes now? And that's because the technologies, we've had the emergence of new metagenomic enabling capabilities, which allow us now to look at this whole system, to think about it in a much bigger picture. We have high-throughput sequencing technologies, computational biology, the genomics, and other omics technologies. What these are allowing us to do is to assess the community composition, its function, and its activity. So we're getting a much bigger picture. The story is becoming much more complex, but that complexity is what we live with as we try to improve crops and how those plants produce. So how do we do this, and how are these technologies enabling us? So you can initially start with a soil sample all the way up to the fruit or the shoots or the roots. You can extract the DNA, so this is one level of complexity that you can look at. And you can amplify that by PCR, amplify a component of the genome, and that will allow you to determine who's there, what's in that community, the species level, across diverse organisms, fungi so far, and microbes, but hopefully in the future, nematodes, whatever else is in that community. Or you can do whole genome sequencing in that using a metagenomic approach and get at what do the genomes of the organisms in those communities encode? What are they capable of? What's their genetic potential for improving the soils or the plants or or inhibiting other microbes? Or what's their potential to cause problems? The other approach is to look at what they're doing in that community, and that's looking at what genes are expressed, and then either the protein or the metabolites that are produced by those genes. And these technologies would involve the metagenome transcriptome sequencing, the metaproteome analysis, and the metabolome analysis. So if we look at it from a simplistic point of view, we can ask now who's there, what can they do, and what are they doing in those communities? Very powerful advances. So I'm going to just give you a few examples of phytobiome studies that have already been published and are perfect examples of how we can use this information and how we can move forward. So this is a study by Ottensen et al., where they looked using a sequencing of PCR fragments at who is there, who's out there in that community. And they did a very detailed analysis of different plant parts. And by just looking at the different colors of the pie charts, you can see that the microbial communities, and this is bacterial communities in this case, are very different on the top of a leaf, from the bottom of a leaf, from the fruit of the plant to the root of the plant. Very different communities that are impacted and that are impacting that plant and are influenced by that plant. They also looked at where those microbes are distributed on the plants, because this can tell us what impact that community might have on that different set of organs. So, for example, if, in the color scheme here, you can see that the Berknea are very abundant on the lower parts of the plant, but not so abundant. I'm sorry, I have it the other way around. Very abundant on the upper parts of the plant, but not so abundant on the lower parts of the plant. Very powerful bits of information when we start to think about how these organisms are influencing the plants. What's really wonderful is we're not limited to prokaryotic organisms anymore. We can now ask these questions with eukaryotic organisms, and this is an example from that same paper where they looked at the fungal communities and where they were distributed on the plants. So again, very different communities on even different upper parts of the plants, let alone in the soil or root regions. In a couple of studies that came out a few years back, they tried to understand the core rhizosphere microbiome and how that core microbiome influences or is influenced by the plant. So what they did is they collected the DNA from the microbes in the soil, in the rhizosphere right around the roots, and then inside from the endophytic compartments of the root. And they asked, what are those populations that are in those different environments? So what did they find? They found that many of the microbes that are abundant in the soil were not found in the endophytic compartment, suggesting there's some selection going on there, as we might expect. And that the endophytic communities from soils were different, from very different parts of the world, were surprisingly similar. So the plant is selecting for communities that are similar from very different root environments. And that the host genotype, in fact, had a role in selection of these microbes. So the bottom line is that these communities are not just products of random assembly, but that these communities are maybe even predicted. So we might even be able, by the way, to breed for better communities. So can we breed plants, for example, that select for a beneficial microbiome? Or can we breed plants that restrict the not so beneficial microbiome? And have we inadvertently, through our breeding processes to date, because we've been selecting for plants on very highly fertilized or highly watered or high tillage soils, have we inadvertently selected away from an ideal microbiome? Lots of interesting questions that I'm going to pose. I, of course, don't have the answers for any of these questions, and that's what the initiative is about, to drive research toward these questions. So you've already seen our wonderful disease triangle once today, and I think all of you are familiar with this. But this is a very simplistic look at what we do as a discipline and the interaction with other disciplines. We need to have a conducive environment to get disease, and we need to have a virulent pathogen. But now what we're learning is that that is just too simplistic, that I like to say we're really talking about genome-genome interactions. And if you think about a cell or an organism is the product of its genome, and how it interacts with other organisms in the environment or in the context of its own tissues, is an interaction of genomes. And those genomes influence what the responses are. And all of that is related to what the environment says it can do. So if we now turn to looking at plant microbe interactions in that context, looking at what is the influence of disease on the phytobiome, the structure of those communities around a plant. This is a study by Trivedi et al., where they extracted DNA from bacteria in the rhizosphere, and then they did two types of sequencing. They did the amplicon sequencing, which enables them to ask who is there, what genus and species are in that community. And they also analyzed the functional genes via a hybridization technique. And that enabled them to say, well, what can those organisms do? It's not saying what are they doing, it's asking what is the genetic potential of that population. And they did this on trees with citrus greening versus healthy orange trees. And so if we come back to the context of genome interactions, they're asking, does the genome of the pathogen affect the genome response of the plant, which then affects the genome content or function of the microbial communities surrounding the rhizosphere? So what did they find? Really very interesting piece of work. They found that the rhizosphere communities on the infected trees were quite different from those communities associated with the healthy trees. That, in other words, disease is associated with detectable shifts in the phytobiome. They also found that there was reduced functional diversity in the rhizosphere communities. So what that says is that in the rhizosphere communities that are associated with the infected trees, I didn't finish my statement there, I guess, with the infected trees, there was less composition of genes that had different biochemical functions. And the relevance of those biochemical functions is that there was a shift away from the use of easily degraded carbons by these microbes in this community to sources that are more recalcitrant. This is consistent with carbohydrate partitioning that's related to photosynthesis. So photosynthesis going to the roots is dropping. So in other words, they are able to, using a phytobiome approach or looking at the microbiome that's associated with this rhizosphere, provide more precise insights into the mechanisms and the processes of disease and the consequences. And by extension, using a phytobiome approach, more able to understand what's going on during resistance, predicting the mechanisms. Furthermore, from an approach that's useful for information in terms of diagnostics of the course of disease, you can identify microbial indicators of disease in the microbiome, or alternatively resistance. So looking at the more complex level is a very nice study by Baker et al., where they looked at the complexity of not only plant communities, so mixed plant communities. They then looked at individual plants and their influence on the rhizosphere. And then they looked at, so that would be plant-plant interactions versus plant-microbe interactions. And then they looked at the impact of a Streptomyces strain that produces a toxic antibiotic compound. So they were looking at what's the influence of that Streptomyces on that microbial community. And while I won't go into the details of that paper, it's very exciting because they were able to show that at different levels, different factors, very complex factors, influenced the phytobiome. So there are many questions that this can lead us to. So why do we care about these things? Well, we care about this because we want to know what factors are influencing the phytobiome content and function, not just the content, but the function of that phytobiome, its ability to be a biocontrol agent, for example, or plant health. And what functions that affect that microbe interactions can be manipulated, for example, to improve soil health or in the health of the plant or to control disease. So many opportunities and many questions that these studies are opening up. We're not just thinking about the phytobiome in the context of plant microbe interactions, but some very exciting studies are emerging now in the insect world. And as we've been promoting this initiative to different societies to get their buy-in and get their voice behind it, what we're finding is that the entomologists got it right away. They see how the interactions with insects and the microbiome are critical to them. So for example, this is a case which was published in PNAS a couple of years ago, where the Colorado beetle actually exploits orally secreted bacteria to suppress plant defenses to the insect so that the Colorado beetle can feed more happily. And if I knew how to activate that video from here, there you would see the larvae secreting the nice microbes and feeding on the plant. So we've seen a lot of different papers emerging now with studies on the phytobiomes and the microbiomes since those early papers. And the field is really heating up, and we're learning a lot about how microbes interact with plants and plants interact with microbes. And that allows us to think about what questions we can address. And clearly, the field of questions is wide open. There are ecological questions that can be asked. There are questions about how the phytobiomes affect plant disease, plant resistance, and plant performance. How do they impact the ability of the plant to take up nutrients? So that impacts those of us who like to eat nutritious plants, and also the animal feed people. They understand that the phytobiome is important for the quality of the food, feed, and fiber that we get. How does the phytobiome impact plant tolerance to abiotic stresses? So if the temperatures go up and down, and does the phytobiome help the plant temper those responses? And from an industrial point of view, what useful organisms or products of those organisms can we mine from the phytobiomes and use in the future? From George's perspective, as the context of this symposia is in soil health, there are many questions that can be addressed from what we heard earlier, that how can the phytobiomes be used to help manage some of those things? And already with the measurements that are being taken, you can understand that there would be different nutritional qualities to the soil. So, for example, could we use the phytobiome or a microbiome to rebuild a depleted soil or to improve a depleted soil if we understood the context or the members of that community? We all hear lots of stories about the human microbiome, and in fact, the Human Microbiome Project has just dominated not only the popular literature, but the scientific literature. And I'm truly amazed by the beautiful artwork that comes with this particular, the human microbiome story. The human microbiome is providing us a nice platform to learn a lot about how we can go forward with understanding the phytobiome of our communities. So what are some of the lessons that we've learned? Well, for example, we've learned that these large-scale consortium-driven projects could be highly effective in learning about microbiomes. And what they've done is they've established not only the infrastructure, but a lot of the technology. Same thing that happened with the Plant Genome Project, which followed the Human Genome Project. The sequencing technologies, the informatic technologies, in this case, the metabolomics, the proteomics, all of that we're going to be able to glean. We don't have to reinvent that. And although the microbiomes exhibit extensive variation across humans, there are some really interesting conserved patterns and profiles that can be recognized. Now, we recognize the complexity that we have more than one host. We have a tree versus an Arabidopsis plant or a soybean, very different hosts in very different environments. We're going to have to adapt in order to glean from that information. The major challenge for the Human Microbiome Project, which we will have to face, is that they're moving from correlative information to understanding causation. And that transition of these longitudinal studies, which are much stronger than the cross-sectional studies, is going to influence how we have to move forward. So the human microbiomes has given us some very interesting paradigm shifts. The commensals are not just passengers. They play critical roles in how our immune systems, our digestive systems, our behavior, and our nervous systems. If we disrupt the homeostasis between the host and the microbiome, we can have, it in and of itself will have a larger effect than the host genetics themselves. And I think that one of the most fascinating things is that the healthy microbiome may not just be a result of the specific set of microbes that are there, but more importantly, the functions that those microbes can carry out. A lot of application, and I'm going to speed up a little bit due to time. You've probably heard about fecal transplantation. This has been enhanced by studies of the human microbiome, and it's established a wonderful foundation for probiotics, the use of probiotics or prebiotics, and the information needed for that. But the phytobiomes, of course, present unique challenges that we haven't seen in the human microbiome. For example, the GI tract is very static in structure, where plants grow continuously. The gut microbiomes exhibit this homeostasis and dysbiosis, but we don't know if the phytobiomes attain a homeostasis, or if even this dysbiosis is an appropriate term for the phytobiome. We know that the gut is a host-dominated environment, but the phytobiomes are influenced by other plants, by soils, by animals, insects, or fluctuating environmental conditions. So our level of complexity is much greater than in the human. So this is why taking this systems-wide approach will give us a much, much more robust approach to understanding, for example, how can we manipulate the phytobiome to better control disease or to better make nutritious and safe plants. And there are many opportunities that we have because we're in a plant system, greater opportunities to look at causal relationships, greater power to identify host genotype effects, greater power to associate the phytobiomes and plant health outcomes, and a greater way to enable rapid application of what we learn to the field, because we're dealing with plants and not with people. So to wrap up, our goal is by 2025 to build a foundation to assess climactic impacts. We need the science behind all this to understand interrelationships with nutrient uptake and their utilization, to relate the phytobiome to its impacts on animal and plant health, to safely and sustainably intensify production of food, feed, and fiber, and to literally change the paradigms for plant disease control and crop improvement. So we have a lot of initiatives, and I think you've heard some of them. We're planning a symposium in 2015 that is designed to bring not only these many disciplines, you have to have microbiologists, soil scientists, animal scientists, ecologists, epidemiologists, bring all those people to the table to lay down a pathway or a roadmap for the phytobiomes for the future. And our goal will be to establish this roadmap, this research roadmap. We also are advertising for an APS Public Policy Fellowship for an early to mid-career scientist to work at a high-level government office and to help us promote the phytobiomes, because the Phytobiomes Initiative is about driving funding for research, education, extension towards understanding what's happening in the plant community. I invite you to visit our booth, and we want you to ask your local representatives and your national representatives to add $100 million in new funding to phytobiomes research so that we can, if you can get these people to work together to do this, we've accomplished a lot in our goal. So contact information here, and I'm going to close with thanking the Public Policy Board members who have helped in all of this effort. And I want to call out Gwyn Beatty, who has been sort of my mentor in phytobiomes and has been very gracious in helping to pull a lot of the thinking together for this presentation and for our grant proposals for meetings. And of course, APS Council and APS Foundation, who are supporting us. Now, here's your job. My job's done. Your job is sitting there. Take out your phones right now. Those of you with smartphones, I know some of you don't have smartphones yet. And tweet your representative. I learned to tweet last night. Tweet your representative. tweet your representative. And this is an example of how to do it. And if you don't know how, stop by our booth, and my very smart young colleagues will teach you how to do it. Thank you very much.
Thank you, Jen, for providing such a background and making us think about the possibilities of plant biome in crop protection. Good afternoon. My name is Dina Arampally. I'm the President-elect of the Canadian Phytopathological Society. On behalf of the CPS membership, I wish to extend thanks to APS for your hospitality at this meeting in Minneapolis. A great deal of thought, effort, and energy goes into organizing a joint meeting like this, and congratulations to the organizing committee for a successful meeting. The Glenn Anderson Lectureship on Security of the World Food Supply Award was established by CPS and APS to provide for special lectures on the security of world food supply. The award recognizes the contributions made by Dr. Anderson, an eminent Canadian agricultural scientist, to the security of the world food supply. In the 1960s, Glenn helped to lead accelerated wheat improvement program in India and Pakistan. His outstanding leadership on research and production effort put bread into the mouths of tens of millions and led to the invention of the term, you heard it, Green Revolution by the press. Later, Glenn went on to train many scientists in this area. This year's Glenn Anderson Lecture is presented by Dr. Jennifer Ann Thomson, a professor of microbiology at the University of Cape Town in South Africa. Jennifer's own research revolves around development of virus-resistant and drought-tolerant maize. However, she has an extensive international involvement in Africa, including the countries South Africa, Kenya, Burkina Faso, and Nigeria. Jennifer is the vice chair of ISA, known as International Service for the Acquisition of Agri-Biotech Applications. This international organization is dedicated to transfer knowledge on agricultural biotechnology. She is the past chair and current member of the African Agricultural Technology Foundation, based in Nairobi, which transfers intellectual property royalty-free from multinational companies to the African farmers. Ladies and gentlemen, please join me in welcoming Dr. Jennifer Thomson.
It's such an honor. And when I looked at the people who've given this lecture before, I'm really awed. But I'm hoping that Glenn Anderson and his mentor, Norman Borlaug, would approve of what I've got to say today. I tried to talk to my slides, but it's a little tricky, so I'll do my best. So, genetically modified crops, that's my field. I work in GM crops, which is highly controversial in many parts of the world. But I'm talking today about healthy plants for healthy bodies. When we think of the role of agriculture in Africa, we must realize that 50 to 75% of the labor force is involved in it. But more importantly, 70% of the population depends on agriculture for their sole source of income. But the real crux and the real problem is that Africa's production is the lowest in the world. And 25% of their grain is imported. That's a huge problem. We've got to handle this somehow. I'm going to skip that. A little while ago, about 5-10 years ago, we had a meeting organized by the United Nations to interact with agricultural specialists from various African countries and come up with a list of priorities. And I don't have time today, and I know we're running out of time, so I'll keep this brief. what are the priorities for Africa that can be handled using genetic engineering? And, whoopsie. Okay, so some of them include, as Dina says, dash it. I'm using the wrong thing. Okay, this is the pointer. Sorry about those on this side. Virus-resistant African crops, including maize streak virus, which is what I work on, and cassava mosaic virus are just some of them. Then another one I'm going to be dealing with is insect-resistant maize. Insect-resistant crops were developed here in America, but we need them for African varieties. So it's not necessary. You can't just import what's developed in other countries to Africa. You've got to develop them on the ground for Africa. And I'm also going to talk about cowpea, not something that many of you know about, but my friends here from Nigeria know all about cowpeas. This is very interesting, decreased fungal toxin levels in insect-resistant maize, and something that we actually thought at the time might not be relevant, might not be doable in the short term, and I'm going to show you that in fact it is, drought-tolerant African crops. I'm going to talk about some of these, so I won't bother about this, but the important thing is we are only going to use a GM approach when other cheaper options are not available. So we're only going to use it if it really is the last-ditch result, or that it produces it better, produces the result. So first of all, virus resistance. Now, I'm talking about maize. Why is maize so important in Africa? Because some people eat maize 3 times a day, but we import nearly 30% of it. And in this case, our target is maize streak virus. I must tell you that there are 2 types. I do talk about this later, but in case I don't, there are 2 types of maize. There's yellow maize, and in Africa, yellow maize is given as feed and fodder. Humans do not eat yellow maize in Africa. White maize is eaten, and unfortunately, white maize hasn't got as many nutrients. So it's a cultural thing, and white maize is a problem in terms of vitamin enrichment, and we'll talk about that just now. Now, here's my field. Maize streak virus, it's obvious why it's called maize streak virus. And this is taken in this small-scale farm here. It's taken in what we call a shamba, which is the Swahili word for a small farm. That I do a lot of my work in Kenya. And he's not going to get much of a crop due to both the virus and due to drought. So what can we do? Oh, and that's what it can look like. Now, I wouldn't be telling you about this if we hadn't gotten it to work. So this was our first result. And this is our control here, eaten by the virus. And here's a virus resistant. They're taken in the lab because this virus is so vigorous that plants hardly get beyond this sort of height even in the field. So we got it to work. I'm not going to go into the I'm a molecular biologist, so I'm not going to go into the molecular biology of it. But this is my brag slide. Okay, maize streak virus resistant transgenics, maize first for Africa, which was indeed what it was. My second brag slide is that we made the cover. But the important slide is the next one. And I'm going to read it to you actually from here because I can read it better from here. GM technology develops in the developing world. The first genetically modified crop developed entirely in Africa is gearing up for field trials. Its success would be a milestone. But what I want you to look at is the date. It's 1997, am I correct? I can't see terribly well. Yeah, thank you. Yeah, I'm not that old. 2007. And there they were saying, that was in Science, they picked up our article and they said what a step forward it would be. Ladies and gentlemen, we have still not done field trials on this in South Africa. Why? Bringing an idea to market is simply too expensive for somebody like me who works at a university, even though I have a partner as a seed company in South Africa. It's not a multinational. It's a small seed company. We cannot afford. This was developed by CropLife International in 2011. 13 years if you're big. Took us about 20. We're little. But look at the cost. 136 million. We don't have that sort of money in Africa. So these things are still sitting in the labs, in our labs and in the labs of our seed company partner, because we simply don't have the money. So when the anti-GMO lobby says it's only the multinationals that are producing it, of course it is, because they're the only people who've got the money. We don't have that money. So we have to go into public-private partnerships. And this is, I'm getting a little ahead of myself, but next month I'll be going to Addis Ababa to speak to the African Union on the importance of research for development. And this is one of the things that I'm going to be telling them, that it's absolutely essential for governments to put money into this kind of research. Otherwise, it's only going to be the multinationals. And in fact, what's just recently happened is that Pioneer has bought out our seed company, and they said, well, actually, Maize Street virus isn't going to make us much money because only it's an African problem. There's no Maize Street virus on any other continent. So here is where we need some benevolence, and where we need some government-private partner interactions. But fortunately, I was born with the persistence fairy at my side, and so we soldier on, as we say. Okay, that's the Maize Street virus. In addition, there's cassava mosaic virus, which is cassava, which is sometimes called manioc, eaten by a lot of people in Africa. There's an international consortium working on this, VERCA, with Danforth and other organizations, and they're doing successful glasshouse and field trials. But again, it's only at that stage. We're not anywhere near commercialization. That's what it looks like. Okay, now let me tell you something about insect resistance, which is linked to post-harvest. I know a lot of people are interested in post-harvest problems. Post-harvest fungal infection. Okay, the crops involved include maize, sugarcane, cowpea, and cotton. And my first slide is showing you it in sugarcane. This is what happens in maize and cotton, where the moth lays her eggs, and then the larvae burrow in and absolutely decimate. You can think of this as a maize, as sugarcane, or as cotton. And so what can we do about it? The solution is a bacterium called Bacillus thuringiensis. I'm sure you know all about this, so I'm not going to go into that. But the beauty is that the Bt toxin targets only insect borers. This is a slide taken from South Africa, where we first started this with, in cotton, which isn't a food crop, but you can make money out as cotton farmers and buy food. I know we're interested in food here. But look at the dramatic differences in the Bt cotton and the non-Bt cotton. That's how the other thing that is so important in Africa is, things aren't on tap in Africa like they are in other countries, in developed countries. And this farmer is mixing a herbicide, an insecticide in a Coke bottle. And you say to him, what are you going to do with that Coke bottle when you're finished? I say, oh, well, we'll just drop it in the ground. And then a kid could pick it up and put water in it, and then what happens? They get insecticide poisoning. So it's very different. And another thing is that, water doesn't come out of taps always in Africa. And so you have to go and collect your water to do your spraying. And who collects the water? The women and the children. Usually the women and the girls. So if we look at the linkage, so obviously insect resistance is important, but let's look at the post-harvest problems with fungal infection. Now, if you're a farmer in Africa, you will store your maize in cribs like that, and they will be rained upon, and they will have sun, and what is more, that's what happens. If the maize has been nibbled by insects that have little holes in it, perfect breeding ground for fungi. And what do we get? One of the things we get is liver cancer. Not very pleasant. One of the other things we get... Oh, now, what I wanted to tell you is if you're a farmer and if you're a farmer in Africa, you're probably a woman. And what you'll do is you'll go to your crib and you'll take out this maize. And you say, I'm not going to feed that to my family. It's pretty awful. What will I do with it? I'm not going to waste it. I'll brew it into beer. And of course, you know what that brewing process will do to fungi. And indeed, esophageal cancer is endemic in parts of South Africa for that very reason. So if we get insect-resistant GM maize, we can also get improved levels of post-harvest health, if we put it that way. And this is the same technology. Now, this is done in West Africa and in Nigeria and places like that, where a lot of cowpea is consumed. And this is an insect that damages the cowpeas. Yeah, they could see the the insects there, and small bites on the transgenics. Now, this is quite interesting, is that, you know, in field trials, you have to make, I mean, it's terribly dangerous. So you have to have everything cordoned off and all these notices up. But of course, in Africa, you know, you've got kids running around, and what does a kid do when they find a fence? They'll climb over it. So if you've got a fence, you've got to guard it. And I mean, these things are so, it's such overkill in terms of regulations that it really is, these are the kinds of things that are stultifying the development in places like Africa. Now to drought tolerance. I know I'm going to lickety-split, but I need to get through things. Now, this is my other field of expertise, and that's why I'm quite proud of it. What we decided to do was to look at African plants that could tolerate dehydration. And this is what we did. We took genes from a resurrection plant. And the reason we call it a resurrection plant is when it's hydrated, it looks like that. But when it's dehydrated, they look absolutely dead. They lose all their chlorophyll. Ladies and gentlemen, if you add water to that plant, within 72 hours, it resurrects. It's like a miracle plant. And they've got amazing capabilities of re-synthesizing chlorophyll and just becoming alive again. And so we figured that, you know, they must have some really interesting genes. So we set about to look at that. And again, I wouldn't be telling this if we hadn't been successful. Oh, but this is the resurrection plant. Often it's found in cracks in rocks, quite close to water. The ones that we found, we had to climb. We had to climb up into the mountains to find them. And they were growing certainly in cracks in rocks. And there they were. Here's a transgenic tobacco. You see, the other thing that I didn't quite understand when I started all this work, apart from perseverance, I've also got quite a lot of lack of knowledge. And I didn't realize that you actually couldn't grow maize in Cape Town. So I started working on this in the glasshouse and didn't realize you could actually not really propagate maize in Cape Town. So hence part of my work is done in Nairobi. But a lot of our work is done on model plants. So here's a transgenic tobacco with just with no water, and there you can see the transgenics and the controls. Here are some better ones. What we actually find now more recently is that if we don't water plants, the controls and the non-transgenics, they both die down. But then if you water them, the transgenics recover and the non-transgenics don't. So that's what this slide shows. Here's the wild type not recovering, and here the transgenics recovering quite nicely. Now, we're not the only people working in this field, obviously. Oh, here's my lab in Nairobi at Kenyatta University, where we are doing some work on the transgenic maize, because they can grow maize in Nairobi, because they sit on the equator, and they actually can even get 2 crops of maize a year, which is really, really nice. Now, this is a very interesting partnership in Africa, Water Efficient Maize for Africa, WEMA. It's managed by, it's managed by the African Agricultural Technology Foundation, of which I'm a member. This is this amazing organization that takes intellectual property from multinationals and delivers it to the farmers royalty-free. And the gene in this case comes from Monsanto, somewhere it says that. There we go. And it's being tested in, CIMMYT is very important. They breed these genes into the right varieties, and then they're taken by the National Agricultural Research Services into the different countries, and we test them there. So there are lots of initiatives going on in drought tolerance. My students often used to say, I know you're chair of the board of the AATF and you go around the world talking about the importance of women, but what about the importance of our project? And I say to them, you know, when it comes to the field, you don't know what's going to work. Maybe the WEMA project with their one gene will work. Maybe our project, we've got 3 genes and we've got an inducible promoter, so they're only switched on under stress conditions. Maybe that will work. Who knows? But I said if we've got more grist to the mill and more approaches, the better chances we're going to have of getting success in different environments. I mean, you're a plant pathologist, you know that everything is different. I mean, Jan was just talking about the differences in different environments. You can't predict what's going to work. You can't predict what works in the lab. You can't even predict always in field trials what's going to work in another situation. I'm moving on now. Bacterial resistance, there's some fantastic work being done in Kenya and Uganda on bananas. Bananas are a major food source in some parts of Africa. And this is work being done by Lena Tripathi and her team under the IITA, which is the International Institute for Tropical Agriculture. And I mean, I think this is amazing. They developed their own transformation system. I mean, I learned transformation from people who knew how to transform maize. They developed it all on their own, sitting in Uganda. Marvelous. And their source, I'm not going to go into this, but their source of genes, this is for resistance to bacterial Xanthomonas wilt. From the sweet potato, I'm not going to go into that, but those are the disease evaluations. They're doing field trials. They've done all this in about 7 years, from the development of from the transformation system to the testing in the field. Amazing stuff being done in Uganda. Now, my final few slides are going to be on the controversy, because I know it's not so bad in America and Canada, but boy, come to Africa and it's really controversial. Sorry. So, you see, the developed world says to people like me, we've got enough food, and we correspondingly say, we don't. We're desperately short of food. So they say we don't need GM crops. We said we need any technology that can increase food availability. I'm not told this so much, but I used to be saying, oh, you're naive, there's enough food being produced. Well, you come to Africa and try the roads in Nairobi and the transportation that's non-existent. I mean, it's fine to produce the right amount of food, but you've got to get it to where it's needed. And this is quite an interesting slide from the ISA organization that I'm vice chair, and Paul Teng is somewhere in the audience. He's the chair. And this is a global area of biotech crops. And you can see how it's taking off. This is the developing world. This is the developed world. This is the total. People say to me, oh, poor farmers, they're forced to buy these seeds. If they're forced to buy their seeds, why do they continue to buy them? I mean, you know, it's because they get the results. That's why we are increasing in the developing world. A very interesting study. Have I got another 5 minutes? Okay, 5 minutes. A very interesting study by a colleague of mine at the University of Pretoria. You see, South Africa is actually the only country, developed country as well, where the GM crop is being produced where people eat it 3 times a day. I mean, you don't eat your corn that's GM 3 times a day. You don't even eat your soybeans 3 times a day. But Africans do eat it 3 times a day. So it's very different. And it's produced by smallholder farmers. So my friend there, he went and looked at the farmers who were planting it. And they cut down, if it's weed resistance, they cut down their weeding from 275 to 150 hours. And the important thing that the people who weed them are mainly women and children. So now they can do other things like go to school. And what interested me is that they asked the farmers, Manus asked the farmers, If you were given a choice, would you go for insect resistance or weed resistance? And I thought, this is a no-brainer. They'll all go for insect resistance. They all went for weed resistance because there's always weed, they're not always insects. And they always have to weed, they don't always have, and of course they can't afford sprays, so they just say, oh, well, c'est la vie, we will just lose part of our crop if there are insects. So that's very interesting. But of course, really rather have both. And in fact, that's what they do. Most of the crops now are both insect and herbicide resistant. But I heard a farmer on the radio, South African radio, being interviewed. There were two farmers, a black smallholder farmer, and they said, why do you plant GM maize? And he says, because I have a profit of 34% in you to that extent. And they said to the white commercial farmer, why don't you? And he says, it's not. What did he say? If there are any Jewish people in the world, it's not kosher. But it's not that. He actually said something, it's just not on. You don't do this. It's not right. So you talked about that to a black farmer. I've forgotten the word. I don't know why I've just lost the term. Anyway. We're talking about food safety issues. This has all been so clear, well documented, that as long ago as May 2004, FAO of the United Nations said no deleterious effects from consumption of foods derived from GM crops discovered anywhere in the world. It's been endorsed by Royal Society, all those. But look at the misinformation. I spoke at the United Nations a number of years ago at the time when Zambia was going through a drought, and the Zambians were refusing food aid because it might contain GM maize, and the farmers who were starving might plant it, and then Zambia would lose their GM-free status for the maize that they were going to export when they could plant maize. The illogicality of this is astounding, but it's scary when you get something like this. When Greenpeace, and this is, see, I got it from Greenpeace, they're saying that GM food will make you infertile. Now say that to a man, that you will become infertile if you eat this. That's a very powerful anti-GM motivation. So I'm afraid misinformation like this is really very stultifying. I don't have time to go into environmental safety issues, but again, the US National Research Council, the FAO, saying GM crops have had fewer adverse effects because, in fact, you get things like reduced and conservation tillage, you get less use of insecticides, etc., etc. So, in conclusion, transgenic food plants are not a magic bullet. They are not going to solve all our problems in the developing world. We need to improve our infrastructure. We need to get better roads and rail transport to transport the food that is produced. We need to educate women. That's the best thing. If you want to feed children, educate their women. So as an educator myself, I'm very much in favor of that. It would be nice to end wars and corruption because that's also a problem. But it would be shameful if well-fed Westerners trade issues, and on the whole, this is a trade issue. The Europeans don't want to get it into their countries because they don't want to have multinationals taking over their food supplies. It's all about trade issues, and they're well-fed, so they don't need it. But it would be an absolute shame if trade issues in the Western societies prevent this technology for helping to feed the poor. And of course, I mean the poor in Africa, because that's where I come from. Thank you very much.
Thank you, Dr. Thomson. Unfortunately, we only have time left for a couple, three very short questions. So for those of you who are watching us via live feed, you could tweet your question at hashtag APS-14, and we are lucky to have Angela Recker sitting here, we'll receive your question, and then we'll ask the panel. So thanks, Angela. Those of you who are here, you could either tweet your questions to Angela, or you could proceed to one of the microphone in the aisles and ask your questions. So here we go. Who wants to be the first? So I don't see anybody. Let's see. If not, maybe I could ask.
Hello. Can I ask?
I don't have a question.
Okay, there's somebody there?
I just want to say thank you. That was.
Please, you state your name and.
Oh, sorry. I'm Margot Bechtel. I'm really short. I'm from Colorado Mesa University in Grand Junction, Colorado, and I just wanted to say thank you. I don't really have any questions. I just want to say thank you to all of you. That was amazing. Thank you.
Hello, I'm Rafi Pearl Travis from Israel. For the first two speakers, I've got the impression that you are embracing now organic agriculture. Is it true? This kind of research, how much does it connect?
Can you repeat the question, please?
From the first two lectures, I have a kind of impression that science will be now embracing organic farming. What do you say about it?
Yeah, I think that's all I got. Organic farming, right?
Well, I can answer that. I mean, organic farming is marvelous if you can afford it, because it costs more. But I can't understand why organic farmers don't go along with GM. What is more organic than a molecule of DNA?
Top that.
Jennifer, thank you for that talk. And you have talked about the role of GM crops in plant health and plant protection. Which countries in Africa are producing commercial GM crops? My second question is, if they're not producing, how do you convince the countries that are not using GM crops?
Thanks. Well, in Africa, South Africa, in fact, 70 to 80% of South Africa's maize is GM. The other major country that's producing is Burkina Faso, which is producing GM cotton. And they are doing it because it's largely been driven by the farmers. The farmers were losing their crops, and they went to the government, and we said, we need this technology, we want it. The neighboring countries, I mean, Mali is one of them, but Mali's got a war, so that's a bit of a problem. But, you know, it has to be driven by the farmers. It has to be needs-driven. You can't drive this and say, you need it. You must let the farmers talk. So Sudan is another one. Sudan is planting. Egypt has. They're not at the moment for reasons I don't quite follow. But if you want to introduce a new technology, it's got to be seen by the farmers as required. And then you've got to have the farmers speak to the politicians. Because in the end of the day, the politicians want to get elected, so they have to talk to their constituencies. So what we're trying to do is to show the farmers through field trials and through extension officers. Extension officers are so important in this area, to show farmers the value of it, and then let the farmers go to the politicians and say, like they did in Burkina Faso, we want this, and we're not going to vote for you unless you give it to us.
Angela, do you have any question?
We do have, so most of the Twitter feed is largely comments on how great the lectures were, but we do have one question for Jan. What is the realistic approach to working with other countries on this phytobiomes initiative?
I think that's a great question. One of the big advocacy efforts is to involve the international community, because microbes know no borders, plants know no borders. So one of the ways that we're trying to do it is by bringing international members onto the Public Policy Board, by reaching out to the scientific societies in the other countries, and asking them for help in this advocacy effort. And when we have these symposia that are coming up, we'll be inviting international speakers because there's a lot of research in this arena around the world.
Okay, there's a question there.
Thank you. I'm Olivo George Mtunge from China Agricultural University, but Tanzanian by origin. Now, in Africa, apart from the use of GM, we have the problem of technology, that even those seed plants that are there, they are not utilized well. Now, how these programs are going to address the problem of technology? And there are some other issues that have been, anyway, misinformed, that these GM are going to perish all the natural seeds that were available, that maybe this GM will be having more side effects. Now, how is the technology going to be addressing this issue? Because we hope that GM development, to develop a plant by GM, most of the countries are considering that this technology is very high for African countries. It might not be relevant for most of the countries to afford how to develop it, how to produce the seeds, how to develop it from when they are produced and to continue producing it more.
I didn't hear most of that. I'm terribly sorry.
How is the technology, GM technology, going to help feed the masses?
How the African countries are going to adopt the production of GM crops. Is it high technology for African countries to afford using it?
You are interested how the technology will help.
How is it going to be adopted by African countries?
Is it affordable? Oh, thanks so much, right. Well, that's the question that was asked to this farmer on the South African radio, because, yes, the seeds are more expensive, but he said, bearing that in mind, with the decrease in spraying of insecticides and things like that, that it's still, he gets 34% profit. So although the seeds are more expensive, the profit is such that it's worth it. But it's not to say that they have to. There'll still be the same varieties available. It's just an added one that is available. But it's not an all or nothing. Farmers can choose. Yes, I want to buy, and they can choose a mix. They can choose ordinary seeds or GM, and they can buy hybrids or open pollinated, and different ones have different. So most farmers will do a mix.
One last question right there.
Hi, Nancy Oshbauer. So I'm on the West Coast in Oregon, which has a lot of organic growers, and something that we're seeing a huge increase in is, I guess the polite term would be snake oil salesmen, where they say that we have all these wonderful soil amendments and all these wonderful biofertilizers that will improve your plant health, and boom, it'll be fantastic. So as this information about soil health and the phytobiomes, as that information becomes available, how do we combat the snake oil salesman?
Can you repeat the last part of the question, please?
As the information on phytobiomes and soil health, as that information improves, how do we use that to combat the snake oil salesman?
How do we use information on the phytobiomes to combat snake oil syndrome? Yeah? Okay.
The salesmen, the guys that are making money off of saying we have all these wonderful biologicals.
So science will help us do that, because I think one of the major influences on making these biocontrol measures work, some of what are called snake oils now may be effective if we understood the soil components, the plant components that influenced how they function. They may function in one soil and not in another. It's because we don't understand what's going on in that community. Some of them are purely snake oil, I agree. But some things may work, but we need to understand that whole community and how it works. It may help us be more reasonable about how we apply chemicals, many reasons. But thank you.
I just remembered what the word was I was looking for. The commercial farmer said it's not ethical.
Okay. Do you have anything about this?
I'll just, actually, I'm going to make an interesting comment here, and that's...
Very short.
No, very short. It's the GMO debate sucks a lot of the oxygen out of the air, out of the rooms, of the general debate that we need to have about feeding the world and feeding people in Africa. So I sympathize with you in that, that there's just so much intense debate out there that other issues, maybe like soil health or fertility or land degradation from erosion, don't really get much attention because people focus on this silly issue and the misinformation around GMOs.
And with that comment, please join me in thanking our speaker for today. And now for our second prize drawing. So you don't want to leave because you may be winning $150 card gift. And you have to be here to be a winner, so hang around. You get to check your tag, the number on your tag. And the number is 099277. 099277. Anybody there? Well, we're going to draw another one then. $203,152. 203,152. Any winner? Well, hang around. Here it comes. 034118. Well, here we go again. Oh, wait a minute. Are you? Well, we have to wait. Are you? You got the number? Well, congratulations. Thank you all, and remember, back to the convention center. The poster huddle is at 3:30, and poster reviewing with the authors at 4 o'clock. Thank you all, and have a good day.